Hi everyone, I'm presenting TRIDENT, a Trimodal Cascade generative framework for drug and RNA-Conditioned Cellular Morphology synthesis. Our motivation is an AI virtual cell, a digital twin of how cells respond to perturbations. As Figure 1 shows, prior work generates only a single modality from the perturbation. We instead use perturbation together with gene expression to drive a cross-modal generation. Existing methods skip the intermediate molecular step. They go directly from perturbation to RNA or perturbation to morphology, but biologically it is the gene expression change that drives the phenotype. So our goal is to exponentially learn perturbation plus RNA to morphology. TRIDENT is a two-stage cascade. Stage 1 is a VAE that fuses the pre-perturbation expression and the drug into a shared latent Z. Regularized to reconstruct the post-perturbation expression, so Z captures the full perturbation effect. Stage 2 is a latent diffusion transformer that denotes image latent, guided by Z through cross-attention. The whole framework is trained end-to-end. To train and test the model, we build MorphoGene, pairing cell painting image with L1000 expression linked by 98 drugs around 98,000 Trimodal samples. We use 44 compounds for training and in-distribution test and hold out the other 54 entirely as the OOD test. On the table, we compare against the MorphoDiff and fine-tuned stable diffusion. In distribution, RFID drops to 49.7, a 5 to 7 times improvement, and KID improves by an order of magnitude. More importantly, OOD FID is still about 3 times better than the baseline, so the model truly generalized to unseen drugs. Visually, the top row is ground truth and the second is TRIDENT. We capture drug-specific phenotypes. For example, the reduced cell density from cytosine B, the two baseline below collapse to a generic monolayer ignoring the conditioning. In LDA space, generated images form tight, well-separated clusters, each matching a specific mechanism of action. For example, store spine and emptying, the model has learned MOA-specific programs. On UMAP, generated and real cells are intermingled on the same main fold and clearly separated from controls, so in embedding space they are essentially indistinguishable. With cell profiler features, the area occupied distribution of generated and real image almost overlap and both shift away from controls, so we reproduce the quantitative morphology, not just the visuals. We also check the intermediate RNA, the predicted and ground truth log fold changes match closely across the 44 compounds with a Pearson correlation of 0.957. As a case study, take docetaxel. Docetaxel enrichment on TRIDENT predicted genes shows downregulation in cell growth and DNA replication, and upregulation in autopotic signaling, exactly its known mechanism. It halts diversion and triggers apoptosis. And the morphology follows. The control is dense and healthy. TRIDENT's docetaxel prediction is sparse dining cells. So the predicted biology faithfully drives the generative phenotype. To conclude, TRIDENT is, to our knowledge, the first framework to explicitly model the full perturbation RNA morphology pathway. Together with our MorphoGene dataset, we achieve up to 7 times better FID, strong OOD generation, and biologically faithful morphologies, a foundational step toward the AI virtual cell. Thanks for listening.